Hi everyone, so welcome back to the disease lessons. So we're now on to lesson 2, which is viruses. So aim of the game today is to talk about how viruses use cells' metabolic pathways and to look at what we mean by antigenic drift and antigenic shift. So your first thing to do is to test your knowledge of keywords and their definitions. So these words will be used within the exam questions and you'll be expected to use them and use other ones within your answers. So on your activity sheet, these should be on there already. So draw some lines to match your keyword with its definition. If you aren't printing the sheet out and you're just answering on paper, that's absolutely fine. Just give the keywords a number and the definitions a letter. So for example, 1A, that sort of thing. So press pause, draw your lines and then come back. Okay, so time for some answers. So hopefully you managed to match the right word to its definition. So pathogen, an organism that causes damage to its host. Disease reservoir, a long-term host of a pathogen with few or no symptoms, always a potential source of disease outbreak. Endemic, a disease which is always present at low levels in an area. A vaccine uses antigens or non-pathogenic forms to stimulate an immune response, which gives protection against subsequent infections. A vector, a living organism which transfers a disease from one individual to another. So, for example, the mosquito in the case of malaria. A carrier is a person who shows no symptoms when infected by a disease, but can pass that disease on to another individual. Antigenic types, different individuals of the same pathogenic species with different surface proteins generating different antibodies. Smallpox is the disease caused by the DNA containing virus variola major. A pandemic, so an epidemic occurring worldwide, crossing international boundaries and affecting a large number of people. Antibiotics, substances produced by microorganisms which can affect the growth of bacteria. A parasite is an organism that lives in or on a host organism. They gain nutrition from their host whilst causing damage to it. And lastly, a toxin, a chemical produced by a microorganism which can cause damage to its host. So tick the ones you've got right and edit the ones that you haven't. So types of viruses. So you can have viruses that contain DNA, that contain RNA, and you can have bacteriophages. Often you'll see just one example in a textbook, and there are several types of viruses with different forms. So if an exam question asks you to draw a virus, they don't want you to draw a very, very specific type. Think of more typical viruses, so the HIV retrovirus, the adenovirus. They're much more typical viruses with genetic material surrounded by a protein coat or capsid and your glycoproteins, so your antigens, on the outside. So how do viruses reproduce is another important one. So viruses reproduce using the host cell's metabolism to copy their own nucleic acid, whether that's RNA or DNA, and to synthesize new protein coats. Some viruses in the lytic cycle immediately reproduce and use the host metabolism to copy their own nucleic acid and synthesize new protein coats. Other viruses may be lysogenic, which means that they'll integrate their nucleic acid into the host cell genome and they'll sit there without having any effect for many cell generations before they begin to reproduce. So how exactly do they reproduce? So if you look at the picture, once the virus has attached to the receptors on the cell, the viral nucleic acid injects into the cell, leaving its protein coat behind. You already know how proteins are synthesized through transcription and translation, but the virus itself doesn't have these organelles, so it has to use the host's organelles to synthesize its nucleic acid and synthesize its protein coats, which they can then assemble to make mature virus particles. So your new virus particles can either cause the cell to burst and release them, so that's cell lysis, or they can bud from the cell surface. The viruses can cause pathogenic effects by cell lysis, by producing toxic substances, by suppressing the immune system, and some viruses even integrate into the host chromosomes and trigger cells to become cancerous. So, as you know, there are many diseases which we can be vaccinated against, and most of you will be familiar with the idea that every year a new flu vaccination is given because the influenza virus mutates. The change in viruses can be attributed to either antigenic shift or antigenic drift. Antigens are proteins which cause the immune system to produce antibodies against the molecule. Antigenic shift causes a much bigger change and results in a virus with a completely novel combination of proteins on its surface. So, for example, the influenza virus has 8 strands of RNA surrounded by a phospholipid envelope with 2 important proteins. Hemagglutinin is the protein that has a role in the virus entering the cell. So I remember that as hemagglutinin goes in. And there are actually 16 different types of hemagglutinin. So H1, H2 and H3 are the most common types in humans. And the second protein is neuraminidase, which helps the virus to leave the cell. There are 9 different forms of this, and N1 and N2 are the most common in humans. So you might have a virus that infects a cell, and you might have more than one virus infecting in different combinations. So for the example that we've got on here, your cell might be infected by the human H2N2 and by the avian H3N8, and then those separate strands of RNA can recombine, which is creating H3N2. And that's actually what caused the Hong Kong flu epidemic in 1968. So that's antigenic shift. So it's a recombination of RNA from different viruses to create a novel virus strain which shows different surface proteins. You also get animal reservoirs, which means an animal virus can mutate and then infect humans as well. So this is why they continually monitor the influenza strains which infect people. Antigenic drift involves very small and very slow changes. So this is easier to vaccinate against. Each time the viral RNA replicates, there's a chance for mutation. And there aren't any RNA proofreading enzymes, so it's very likely that there'll be some form of mutation and that will gradually change the surface proteins. So another diagram just to help show the difference. You can see in the antigenic drift that the tiny changes on the hemagglutinin, whereas on the antigenic shift diagram, your RNA has combined from virus A and virus B to create a novel combination and a completely new virus C. So activity 2 is your virus questions. So press pause, complete your virus questions, and then press play so that we can go through the answers. So, viruses have been described as intracellular parasites. Why is this not strictly true? So, intracellular means inside the cell, which is true. Viruses reside inside the host cell. But remember with the definition of a parasite, an organism that lives in or on its host, obtaining its nutrition from its host while causing it harm. So, yes, your virus is living in its host cell and causing it harm, but it's not strictly obtaining nutrition from a host. For virus structure, you've hopefully drawn something like this, and so in the top right, and you've labelled the genetic material as either DNA or RNA. You've got a protein coat, capsid, and you've got your glycoproteins on the outside. The difference between a virus and a retrovirus, structurally, the differences include nucleotide type, so DNA or RNA, the enzymes that you've got present, so integrase enzymes or reverse transcriptase and integrase, and how they reproduce within the cell. So virus DNA integrates into the host cell DNA, whereas viral RNA is reverse transcribed into DNA and then it integrates into the cell. So looking at how your virus infects your cell. So in this case, you've got a retrovirus, so RNA containing. So firstly, the virus injects the RNA into the host cell. Reverse transcriptase uses the RNA template to make the DNA. The HIV DNA inserts itself into the host cell's DNA. You then get transcription. So the viral DNA codes for viral RNA and proteins. It makes new viral RNA and proteins, which then bud off, and the virus particles split off from the host cell. So all of this could cause harm to your host organism through cell lysis, through creating toxins, suppressing your immune system, or triggering some cells to become cancerous. Hemagglutinin and neuraminidase, so remember hemagglutinin helps viruses to get in. It's the surface proteins that bind to receptors on cell surfaces and enable the viruses to enter cells. Neuraminidase is surface proteins that enable viruses to leave cells. Antigens are toxins or other foreign substances which induce an immune response in the body, so especially the production of antibodies. So antigenic types are different individuals of the same pathogenic species with different surface proteins. So you have your two processes. So name, describe both of these. So figure 1 was showing you antigenic drift, mutations in your RNA or DNA occurring during viral replication. These mutations correspond to gradual changes in your surface antigens over time. And this occurs because there's no proofreading enzymes being present to check for those mutations. Antigenic shift was figure 2. If a cell is infected by viruses that have different combinations of antigens, the strands of DNA or RNA can recombine in a different way in the new virons. So if human and avian influenza infect the same cell, a new influenza can be produced. An antigenic shift is how bird flu, swine flu and COVID-19 were formed. So identifying which statement matches each type. Changes in surface proteins due to point mutations are antigenic drift. Where one cell is infected by viruses that have different combinations of hemagglutinin and neuraminidase, antigenic shift. The strands of RNA can recombine, giving rise to new viruses, is also antigenic shift. And no proofreading enzymes being present in antigenic helps to cause antigenic drift. So lastly for this lesson, you've got some exam questions. There are also some more videos on antigenic shift and drift in the extra information links if you would like them. There isn't a video for lesson 3, so for next time, just click the activity sheet and all your instructions are on there.